The overall goal of the following experiment is to assess the sensitivity to ethanol sedation in fruit flies. This is achieved by placing flies in vials and exposing them to ethanol vapor. The second step is to determine the number of flies that are sedated in each vial at recurring time points until all of the flies are sedated. Next, the time point is interpolated when 50% of the flies are sedated for each vial. The results show an index of the time required to sedate fruit flies with ethanol.
Demonstrating this procedure today will be Simran Sandhu, a student in my laboratory.
To begin the experiment, collect flies in fresh food vials in groups of 11 of a single sex for up to 5 minutes of CO2. Allow the flies to recover overnight in food vials in an environmentally controlled space. Prepare ethanol solution by diluting pure 100% ethanol in purified water to a final concentration of 85%. Allow the solution to return to room temperature overnight. Before the assay, prepare a clean, empty food vial which will serve as the testing vial, a new cellulose acetate plug, a silicone stopper, and 1 milliliter of ethanol solution for each vial of flies to be tested.